Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different properties of the enzyme in the course Enzyme Science and Technology. In this particular module, we are discussing about how you can be able to purify the protein from the cells which are overexpressing the particular clone, right? And what we said is that protein is providing you the different types of options. You can be able to use the charge, hydrophobicity, or the surface area as a criteria to purify the protein. And in the previous lecture, we have also discussed about the basic principle of the affinity chromatography and how you can be able to use that for purification of the proteins. If you recall what we have discussed about that the affinity chromatography relies on the interaction between the receptor and the ligand, and you have the option of either putting the receptor along with the protein or the ligand with the protein, and that's how you can be able to use them for the purification of the particular protein. So in a typical affinity chromatography, what you have is you have the receptor and the ligand, which are exclusively been interacting with each other, and you have the option that you can actually be connect the receptor to the matrix, right? Or you can actually be able to connect the receptor to the protein. So either of those cases, the ligand has to be placed on the counterpart. For example, if you are putting a receptor onto the matrix, then the ligand has to be placed onto the protein of your interest with the help of the recombinant DNA technology. If you're putting the receptor onto the protein, then you have to put the ligand onto the matrix. So we have a couple of examples which we are going to discuss in this particular lecture. So first thing what we require to perform the affinity chromatography is that you should have the receptor or the ligand. The receptor molecule present on the matrix can be produced either by the genetic engineering or the isolation from the crude extract, or in the case of antibody, it is produced in the mouse or rabbit models. So you have the three options. You can actually be able to purify the receptor either with the genetic engineering, or you can isolate the receptor from a crude extract, or you can actually be able to generate the antibodies. How you are going to generate the antibodies? So for generation of the antibodies, what you have to do is you have to first prepare the antigens. If you are dealing with a protein or if you are dealing with a molecule which is very small and it is coming under the category of hapten, then you have to convert the hapten into an antigen. After that, you have to prepare the antigen for the injections, and after that you have to do the immunization to the animal. You have to inject the animals, right? And then once the animal are actually going to produce the antibody, you have to test the presence of antibodies in the animal. And once you see that the antibody is in a very high quantity, then you can be able to extract out the blood, and that blood is going to contain the antibodies. And from the blood, you are actually going to collect the serum, and the serum is going to be, and then you can be able to verify the antibodies from the serum. So let's see and discuss some of these steps individually. So the preparation of the antigen. So the antigen required for the development of polyclonal antibody is approximately 2 milligrams. It is required for the multiple injection to induce the boost immune response. So it has the following steps. First, you are going to produce the antigen. So you can actually be able to use the recombinant DNA technology to clone the antigen of your interest, and then you can be able to overexpress that in a large quantities. Then you can do the isolation of the antigen. There are two approaches to isolate the antigen from the E. coli expressing cells. Uh, Purification of antigen under the native conditions. So you can use the column chromatography, you can use the ion exchange chromatography, or any other affinity chromatography to purify the antigen. Or you can actually be used the antigen under the denaturating conditions. So you can do the electroelution of antigen from the SDS page. These are the steps what you have to take to isolate the antigen from the SDS page. So first what you are going to do is you are going to run the antigen on the SDS page, followed by you have to identify the region of interest, right? So you are going to identify the region of interest. Then you're going to cut the protein bands and you are putting into a dialysis bag. And this dialysis bag you are going to keep into a horizontal apparatus. and then you are going to run the you are going to perform the electrophoresis, and as a result what will happen is that the protein band is going to be electroeluted uh, from this acrylamide block, and it will come into the dialysis bag, and then you can concentrate the antigen and you can use this antigen for the injections into the animal. But you cannot inject the antigen as such. You have to prepare the antigen for the injections. So preparation of the antigen for the injection, you have to combine 100 microliter of antigen, which is approximately around 100 to 150 microgram, with an equal amount of fluids complete adjuvant to a final volume of 200 microliter. You mix thoroughly to obtain the emulsion using a syringe or a pipette. After 4 weeks of injection, inject the second booster dose. Repeat the booster injections 4 to 5 times every 4 weeks to generate a robust immune response and development of the memory B cells. Then once the antigen is ready, then you can do the in vivo immunization of the rabbit. So before immunization, you can just take out some amount of blood so that it is actually going to serve as a control, and it is called as pre-bleed or the pre-immune serum. And then you can inject the 200 microliter antigen into the rabbit, which is going to be the NZ strain. So this is the rabbit what you have. This is the NZ strain rabbit. And during this step, either use a helper to hold the rabbit or use a restraint device to hold the rabbit. Inject the antigen onto the back of in the rabbit in the form of the buttons. So you're actually going to inject the rabbit somewhere here in the form of the buttons, and these buttons are going to release the antigen in a very slow mode. Then you are going to do the booster. So exactly the same way you are going to prepare the antigen, except that it's here you are going to use the incomplete adjuvants, and that's why you're going to put the boosters. Once you do the booster, you have to wait for the 4 weeks, and that's how it is actually going to have the a robust immune response, and it is actually going to produce large quantity of antibodies. Then you can determine the antibody titers. So you can use the indirect ELISA to determine the antibody titers. And then once you see that the antibody amount is very high, you can collect the blood and prepare the serum. So take out the 20 to 30 ml of blood from the ear vein, or large quantity of blood can be drawn after the cardiac puncture. And you can actually be able to collect the blood, and then you can actually be able to prepare the the serum, and you can actually be determine the concentration of antibody by the indirect ELISA. Then how you are going to couple the receptor to the matrix, right? So once the receptor is being available, it can be coupled to the matrix by the following steps. In the step 1, you are going to do the matrix activation. In the step 2, you are going to do the covalent coupling. And in the step 3, you are going to do deactivate the remaining active groups onto the matrix. So you can have the multiple options. You can use the epichlorohydrin mediated receptor coupling. So in the epichlorohydrin activates the polysaccharide matrix by adding the oxirane groups with 3 carbon alcohol groups spacer arm. The activated matrix reacts with the receptor containing primary amine or thiol groups. Receptors are coupled to the matrix by a thioester or secondary amine linkage. It can be able to couple the hydroxyl group containing receptor molecule as well as by the ether group. So in a polysaccharide matrix, when you add it to epichlorohydrin, it is going to form the activated matrix. And on this activated matrix, when you are going to add a receptor, which is going to have the free amino group, it is going to get tagged, and that's how you are going to have the affinity matrix. Then you can also use the carbamides mediated receptor coupling. So in the carbamide receptor coupling, carbamides reacts with the receptor containing carboxyl group to form the isourea ester and the activated matrix, and then allowed to react with the receptor molecule containing the carboxyl or the free amino groups. The receptors are coupled to the matrix by a secondary amine linkage. So this is what has happened, right? Carbamide, when it reacts with the with carboxyl group, it is going to form the isourea esters, and when it reacts with the receptor, it is actually going to form the affinity matrix. Once you have the affinity matrix, you can actually be able to do the affinity chromatography. So in affinity chromatography, what you have is you have the equilibrium. So affinity chromatography material is packed in an equilibrate with a buffer containing high salt to reduce the non-specific interaction of the protein with the analyte. Then you can have the sample preparation. The sample is prepared on the mobile phase, and it should be the free of suspended particle to remove the clogging of the column, and so on. And then you inject the sample with a syringe. In the elution, there are many ways to elute analyte from the affinity chromatography. You can increase the concentration of the counter ions, or you can change the pH or polarity of the mobile phase, or buy a detergent or the creotypic salt to partially denature the receptor to reduce the affinity for a bound ligand. And once you're done with the elution, you have to also do the column regenerations. So after the elution of an analyte, you can affinity chromatography require a regeneration step to use it for the second time. Column is washed with the 6 molar urea or gonadinium hydrochloride to remove the all non-specifically bound proteins. The column is then equilibrated with a mobile phase to regenerate the column. The column can be stored at 4 degree in the presence of 20% alcohol. So this is what you have, the affinity column, where you have the antibody or the receptor tagged. So when you add the sample, the receptor is going to have the affinity only for the one molecule, right? One of the ligand. So it will go and bind to this ligand, right? And the rest of the molecule are going to be washed away in the washing step. And then you are going to have the receptor ligand onto the column, and then you are going to do the elution. So you're going to have the different options of the elution, and that's how you're going to monitor this. So what you see is that the molecules are getting eluted in the elution step. So bioaffinity chromatography is a step where you are actually going to do the affinity chromatography. So okay, so if you want to do a bioaffinity chromatography, you also going to have the you should you can use that for even for the antibody purification as well. I'm sure we have seen how we can be able to produce the antibody in the serum. So if you see, all those procedures are very complicated. So to explain you those procedure, how you can be able to produce the antibody, and the crude antibody is going to be present in the serum. We have prepared a small demo clip, and that demo clip is actually going to explain you all the steps what we have just discussed, how you can be able to prepare the antigen, how you can inject that into the animals, and so on.
I am Amog Aransasdubite. I work in CSIR, CDR, I don't know. And in today's demo, we'll be discussing different steps involved in generation of antibodies in rabbits. So for the first step, we require several things like trans complete adjuvant. Here it is from Sigma. We need a microemulsifying needle which has two openings connected with a fine needle. We need antigen which is purified and filtered, so there are no contaminations. It is a sterile solution of antigen. Then we take out some of the Franz complete adjuvant in Nippendorf and then mix them together. Since this adjuvant is oil based, it doesn't mix easily with the watery system like the antigen is in previous. So therefore we mix them vigorously and forcefully. For that purpose, we take these two, we mix these emulsion and we mix these PBS containing antigen and the adjuvant, oil-based adjuvant. After mixing them, we take out in a needle. Using a needle, we take out in a syringe like this, and then we fix the microemulsifying needles into it. Attach another syringe into it. Like this. So once you have filled your antigen and the adjuvant in this needle, you push it here, and then you keep pushing from one side, keep pulling from another side, keep pushing from one side and keep pulling from another side. So this process forcefully pushes your material, I mean the oil and the antigen, through this fine needle, and with that, in that process, the emulsion is formed. Emulsion can be called as water in oil or oil in water because both are in the same concentration, same volumes. So you can call them anyway. So the emulsion, by this method the emulsion is formed. So for ready reference, we have already prepared the emulsion. This emulsion looks like white. Initially it was two phase and then slowly it has turned into single phase. Now you can push this emulsion from one needle to another side and from another this syringe to another syringe. So this process creates very good emulsion which doesn't separate out later on when you are ready to inject. So how do you check them? So for checking purposes, we drop one of this emulsion, or drop a drop of emulsion on a water surface like this. If emulsion is not formed perfectly, this will spread out. Otherwise, it will not spread. So this is a check that your emulsion is formed correctly. So once you find that this drop is not spreading, your emulsion is actually ready for injection. So this was the process by which we prepare the emulsion for injection. So this is the first step of preparing the emulsion. So now let us understand why we prepare the emulsion. We have checked that emulsion is formed. Now the purpose of making the emulsion, because you have antigen and antigen, through antigen you can raise antibodies, but after emulsifying them, you actually make the antigen releases slowly. So it is a sustained release kind of preparation, so that the antigen is exposed to system in a systematic manner, so that more and more memory cells can be is generated, and that's the sole purpose of having emulsion. Otherwise, if you inject antigen as such in PBS or in other watery system, it will spread out in the body and it will be cleared out by the immune system readily, and no memory cells will be generated. So this is the main purpose of preparing the emulsion. So now we have prepared the emulsion. We have come to animal house. This is our rabbit, which will be immunized. And before immunization, we have to take pre-immune bleed so that we can compare later the serum and the anti-serum. So we will not start how we immunize it. So now we are preparing to immunize. The first and important thing is in all these animal processes is we have to avoid the pain to the animal. So for that purpose, we have to strain the animal because we have to inject. So we strain the animal in a way that it has less and less pain, and the movement is also less. So we'll inject this into the emulsion into the thigh. We have to catch hold of both the legs. Then we have to sterilize the area using alcohol. We have to look at the thigh muscles. They should be cleanly visible. Skin should be cleanly visible. There are two kind of injection that we give. One is intradermal and another one is subcutaneous. So today we'll be doing subcutaneous injection. This is our emulsion that we have prepared by microemulsifying needle. We have seen earlier. This is the area where we would like to inject. We have to take out all the airs from the syringe and the needle. We have taken out, clean the area again, and then we apply some antiseptic powder. Here it is betadine powder, so that the infection cannot develop later on. We sprinkle some of it at the area of injection. And then slowly release, leave the animal relaxed, and it is immunized. So this is a button form which is made. This is the region that the emulsion is lying. So when we have to bleed, we have to strain the animal, because you cannot, in general you do not, it is not advisable to anesthetize the animal. So we use this strainer, this wooden strainer, where we keep this rabbit here, keep his neck here inside and the head outside. Then we strain his neck using this. Then we strain the rabbit from back side using these kind of cassettes. According to the size of the rabbit, there are 3 slots given. And after using this, we strain the animal from the top, like this. Like this. And we lock the neck region, and the animal is ready with his head out and the ear outside, from where we have to bleed. But the drawback of this strainer is that if the rabbit is smaller or very, depending upon its behavior, if it is panic, sometimes it rotates itself, and the backbone is broken. Its rabbit's backbone is very sensitive, and it breaks. So if it breaks, you have to sacrifice the animal unnecessarily. So we do not use this, though it is used for rabbit breeding, and we normally do not use it. Another method is to strain the animal. So another method is to strain the animal in a towel or this kind of cloth. So the advantage of having this cloth to strain is the animal has its claw outside of this cloth, and then it cannot move. So we have to restrict the movement. Pain is similar in the strainer and on this cloth. So we use this thing. Now we will strain the animal on this cloth. We will keep the animal relaxed on this cloth, and we will strain it. Make sure that the ear are outside and the animal is strained properly, so as to reduce its movement. And now it is ready to bleed. We will bleed the animal from this mid-ear vein, and we rub it so that it gets heated up and the circulation is faster. The vein will also expand and more flow will be there. So this is the method which is normally applied. When the vein is properly visible, this is the mid-ear vein from which we will bleed. We will slightly sterilize it using alcohol and using a 20 gauge needle, which is wide enough to give sufficient bleed. We'll tip the vein and collect the blood. Then to stop it, we just with less and less pain, you can collect the bleed like this. Now we have to make sure that no further bleeding occurs. Then we wipe out whatever bleed is outside. Using sterile water, we wipe out all the blood here and there. If any spillage, we'll wipe out all over using water so that the vein becomes cool and gets shrunk. Then just check, still it is bleeding, so keep it pushed. Until the bleeding stops. So now I think the blood has stopped coming out, and then now we apply some antibiotic. Here in this case it is betadine powder, so that there is no further infections or inflammation in the rabbit, and this also ensures that this is your pinna, and if there is any inflammation, it will have some pain. So it will avoid that kind of pain also. So we have isolated approximately 10 to 12 ml of blood. This will give us approximately half of the volume of the blood serum. This will be coagulated. The blood will be coagulated at 37 degrees centigrade for 1 hour, and then it will be kept at 4 degrees centigrade for overnight, so that the clot is shrunken properly and the serum is maximally taken out. And then we'll isolate the serum, add some preservative like sodium azide, and keep it at -20 or -80 as per the requirement. And then we also taste simultaneously the titer of it and the specificity of it using ELISA test and the resting blood test respectively. Now you can see this whole of the animal. This animal is very relaxed. It has, I think, it has very less pain or a negligible pain. And this is actually very important for handling when you handle animal. That in whatever procedure you go through with animal should be ensured not to have pain. Maybe you can, if the procedure is painful, you understand it is a painful procedure, you anesthetize it. Since this procedure is not painful, we have not necessary. So that this is very important step to ensure that the animal has pain. It should be relaxed. So in whole of this process, I think you have got to know all the steps of embryo development in rabbit, and we prepared emulsion, we injected the emulsion, we isolated the blood after giving sufficient booster doses, and whole of the process is in the I think you have understood most of this process and you liked it. Thank you.
Once you collected the serum in that particular step, it is actually going to have the crude antibody, which means it's going to have the antibody and all other proteins. So you can use the bioaffinity chromatography and you can be able to use that for affinity chromatography. And for antibody purification, what you can do is you have to first couple the antigen onto the beads, right? And that's how you're going to have the antigen bound saffrose beads, and then you can be able to load this particular serum as a sample, and that's how you can actually have the washing step, then you can have the elution step, and that's how it is actually going to give you the purified antibodies. So antibody is how you can actually be able to prepare the column, right? So what you can do is you can use the CNBR mediated receptor coupling. So CNBR mediated coupling is more suitable for the antibody to the polysaccharide matrix such as agarose or dextran. CNBR reacts with the polysaccharide at pH 11 to 2 to form the reactive cyanide ester with a matrix or less reactive cyclic imidocarbonate group. Under alkaline conditions, these cyanogen esters react with the amine groups on the receptor to form the isourea derivatives. The amount of cyanide is more in the agarose, whereas this and that. The protein or the peptide ligand with the free amino group is added to the activated matrix to couple the receptor for the affinity purifications. So this is what you have. You have a polysaccharide matrix. When it reacts with CNBR, it is actually going to form the cyanide esters and as well as the cyclic imidocarbamide, and that is going to react with receptors to give you the isourea derivatives, and the receptor is going to coupled onto the matrix, and this coupled matrix can be used for the purification of antibody, or it can be used even for the purification of the antigen as well. So in the antibody purifications, you can just pack the column with a receptor onto this, and you can incubate the affinity column with a high salt containing buffer, so that it can reduce the non-specific interactions. Then you can wash the column with the 10 column volume using the equilibration buffer, and then you can do the elution. So you can use the counter ions, or you can use the pH of 2, or you can use the detergent or tartaric salt to partially denature the receptor to reduce the affinity for the bound ligand, or you can use also the beta mercaptothenol or DT2, and all of these are going to release the antibodies from the matrix, and that you can actually be able to collect and verify. Uh, Once this has been done, you can actually neutralize the acidic elute with the help of the 1 molar trace containing PS 7.2 containing 150 millimolar. And once you are done with the purification, you have to do the column regenerations. Then we also have the another examples of the bioaffinity chromatography where you can use the GST based bead based purifications. So glutathione GST utilizes glutathione as a substrate to catalyze the conjugation reaction for xenobiotic purpose. The recombinant fusion protein containing GST as a tag is being purified with the help of the glutathione coupled matrix. How you're going to do that is that you are going to prepare a column which is having the GST, right? So that's how the protein is actually going to contain the GST tag, and the column is going to have the GSH as a ligand. So when you load the lysate, it is actually go and bind because there will be an interaction of the GST and GSH, and then you're going to have the washing step, and ultimately you are going to use the GSH for elution. So GSH is going to compete for the protein, and that's how the GSH will bind to the GST, which is already been tagged with the protein, and that's how the protein is going to come out. After this step, you have the option either to remove the tag, so that also you can be able to do with the help of the different types of proteases. Then you could also use the another affinity chromatography, which is called as metal affinity chromatography. So in a metal affinity chromatography, we are going to discuss about the nickel NTA affinity chromatography. So first, what you have to do is you have to prepare the column with the help of a step which is called as charging steps. So the nickel NTA, what you are going to get from the vendors is the plain columns. Okay, so you're going to get NDA agarose or the saffrose beads. Then you within a charging step, you're going to first load the metals onto these beads. Then you're going to load the sample, and then you are going to have the washing steps. And once the washing step is over, then you can do the elution. You can have the multiple options. The mostly people are always using the imidazole the uh, as the ligand for competition. So you can, when you do the imidazole, different concentration of the imidazole is actually going to have a competition with the histidine, which is bound to the nickel, and that's how it is actually going to help you to elute the column. Once you've done it with this, you can actually regenerate the column with the help of washing the column with a high salt concentrations, and that's how the same column can be reused again and again. So we have prepared a small demo clip to explain you how you can be able to perform the nickel NTA affinity chromatography.
For protein purification, first we have to inoculate the culture into this uh, larger volume of ELB class. Then we will induce it. So first I will show you how to inoculate. This is the single colony grown overnight culture. So we can use for the inoculating into large cultures. So this process should be done in aseptic condition. So that means we have to use laminar airflow for this purpose. So, and also we have to remember we should include suitable resistant marker like ampicillin or kanamycin, this kind of antibiotics, or this is depends upon what vector, what resistant vector you are having. So in this case we are using ampicillin as a antibiotic. So let's start. We have lysed the cells using sonication. Now we have to centrifuge the lysate to get supernatant. So that supernatant we load onto nickel NTA column and purify the protein. So I will transfer into 50 ml centrifuge tube, then I will centrifuge. While the centrifugation is going on, we have to wash the column using first, this is in 20% ethanol. So we have to wash first with water, then equilibration buffer. So let it drain completely, that 20% ethanol. Then we will add water, double distilled water. So at least 5 column volumes of water should be added to remove completely. And next we will equilibrate with the lysis buffer, the buffer which we used for the lysis of the bacterial cells. Before equilibration of the column, we have to charge the column. Nickel NTA, there are 2 types of bits are there. One is already readily charged bits which comes from company, and another one is we have to charge. They will give only NTA agarose bits. So here what we will do is we will charge the beads with the nickel and then we will equilibrate. We already washed the column with water and 0.2 normal NaOH again with water. So now we will equilibrate. So this is a nickel hexachloride solution. So we will keep in this condition at least 20 minutes to charge the beads. After that we will remove nickel NTA. We will elute the nickel nickel solution, and then equilibrate with the lysis buffer. So after 20 minutes we eluted the nickel solution. Uh, Next we will equilibrate with the lysis buffer. We have to wash at least 2 column volumes to remove any free nickel which exists in the beads. So after equilibration, next step we will load the lysate and then incubate for binding. This is a paraffin. I am going to close this plug in the pan. So once column packing is over, we kept it in ice and we'll keep in this condition for at least 2 hours for binding. So that histalled protein will bind to the nickel NTA, and in further steps we will elute the protein. After incubation with beads, we have to follow another 3 steps to get completely purification done. First step is we have to wash with the equilibration buffer. First, after the beads taken out from ice, you have to remove the outlet so that all the flow through other than beads will be taken out. And the next step is we have to wash with the equilibration buffer, and the third step is we have to elute the sample, elute the protein, histalled protein using imidazole containing buffers. For all these buffers, the pH should be adjusted prior hand, not like you have to first you take the buffer, lysis buffer, and you have to add imidazole. It's not like that. It may increase the pH of the buffer. So after compiling all the lysis buffer with the imidazole, then we have to adjust the pH so that throughout the procedure the pH won't change. So this is a flow through. Whatever we are getting is flow through. In next step we will wash with the lysis buffer. In this step we are going to wash with the lysis buffer or equilibration buffer. So just this is the lysis buffer. Before doing this, we have to observe the beads. We should not directly load onto beads. You just have to pour through corner, through the wall of the column. Otherwise, it may disturb the beads. So protein may also degrade. So this we have to keep in mind while doing this washing. While doing purification, we have to remember that every time you are introducing new buffer, that time you have to collect the fraction, and this can be used for the running SDs and testing the purity of the samples. And also the flow through part and the washing part, what we have collected, we have to keep it safely. After verification of the gel, only we have to throw. Say you are getting only 10% of the protein in your purified fractions, and 90% of the protein eluding in the flow through. That time you can reuse the flow through for purification again, purifying the protein again. So you have to collect the fractions in a small microcentrifuge tubes, and we have to save those fractions, label it and save. So we washed with the equilibration buffer and we also collected the flow through. Now it's time we will wash with the 20 millimole of imidazole. So this will remove any non-specific proteins binding to the beads. So we will wash with the 20 millimole imidazole containing buffer. Then we will subsequently elute in 250 millimolar imidazole containing buffer. In final step, we are going to elute with the 250 millimole imidazole containing buffer. So what we are going to do is we have to incubate beads with this buffer for some time and collect the fractions. Now we have to collect the fractions. After editing the complete fractions, we have to wash the column with water, then 0.2 normal sodium hydroxide solution, then again water. After final wash with the water, then we have to store the beads in 20% ethanol. So I will wash it and store it in the ethanol. While the washing is going on, we have to take 50 microliter of each fractions and run it on SDS phase. That will give the purity of the fractions. We have to heat the samples before loading onto SDS phase, and also we have to keep all these fractions what we have collected at 4 degrees Celsius for further confirmation of the purity. Once the purity is confirmed, we have to dialyze those fractions against our buffer of interest, then use for the further studies. So we purified the protein using nickel NTA column. We run the gel and stained or de-stained. So now it's time to document the gel. So we have to identify whether we got any single band fraction or not. So this is the gel I kept on white tray. Now just close it. So we have loaded marker and this from this side. Second one is the load, This is flow through, wash 1, wash 2, and these fractions are eluted fractions 1, 2, 3, 4, 5 serially. So as we can see, uh, the eluted fraction showing a band corresponding to this protein, uh, but the molecular weight can be calculated using the software, image lab software. So as we can see, in the protein corresponding to this purification, his tagged one, it is going most of the fraction in the flow through. So we can use, as I said in the video earlier, we can use this flow through action again for purification of the protein. You can incubate this flow through with the same beads, and you can repurify again. So that will increase the productivity getting the protein. So these are all other bands whatever we are seeing in the protein, this eluted bands, those are because of the contaminants or degraded protein. Contaminants sometimes may come because of histidine, 3 or 4 histidine having in folded state, that will give possibility to bind to nickel NTA column. And also washing, vigorous washing should be done. If you don't wash properly with a high amount of imidazole, that will give you this kind of non-specific binding. So with this, we'll conclude the video. So I hope it will help you to help you in your work for during protein purification, or help you to understand how protein purification works. Thanks for watching.
So this is all about the different chromatography technique what we have discussed. We have discussed about the ion exchange chromatography, HIC, affinity, and we have also discussed about the gel filtration chromatography. Now the question comes, how you can be able to use these different chromatography techniques for purifying the proteins or the enzymes. So we have taken a couple of examples to explain you how you can be able to utilize these different chromatography techniques for the purification of the enzymes. So first example is that how you can be able to do the enzyme purification from the plant sources. So purification of the enzyme from the plant sources. So we have taken the example for the plant, which is the Nigella sativa. And you are going to do the extraction of the substance or the protein. So you can follow the scheme. So you can prepare the water extract of the N sativa, and you can dry it and make the powder. So powder is going to be dissolved in PBS 6.4. So that is actually going to give you two fractions. It's going to give you the insoluble pellet, or it is going to give you the soluble extract. So within the soluble extract, you're going to have the oily layer or the soluble extract. This soluble extract is actually what you're going to use, and you're going to dilute that against the the 0.05 molar phosphate buffer using the 3500 molecular weight cutoff, and as a result it is actually going to remove all the contaminating substances, and you can actually be able to estimate the protein concentration with the help of the Bradford, and this is what you're going to use in the chromatography. After this, you're going to perform the ion exchange chromatography. So you're going to use pack the column with a DA saccharose, and you're going to then elute with an NaCl gradient at pH 6.4, and you can do the detection of the protein at 280 nanometers, and the analyte band protein ranging from the 94 to 10 kDa you can actually be able to use for the monitoring the proteins what you have isolated from the plant sources. Then you can also, we have taken another example where you can actually be able to use that for the animal sources. So purification of the protein from the animal sources. So we have taken the hen egg. So first you're going to do the extraction. So what you're going to do is you take the fresh egg which you are going to collect it. Then you do the isoelectric precipitation of the egg white with the help of the 100 millimolar NaCl. So that is going to give you the two proteins, that is ovomucin and the precipitate. This precipitate you can just dissolve into the 500 millimolar NaCl with a stirring at 4 degrees overnight, and that is actually going to give you the some amount of precipitate and the supernatant, and that supernatant you can use for the chromatography into the further use in the chromatography. The supernatant you can load onto a Q-saffrose anion exchange chromatography, and followed by the sp saffrose, which is the cation exchange chromatography. So remember that in the previous example we have only used the single column of the ion exchange chromatography. We have used the two column in tandem actually. So isocratic elution of the sample using the 0.4 1 molar solution, followed by the gradient elutions of the NaCl from the anion exchange chromatography. The unbound fractions were collected and used as a starting material for the cation exchange chromatography. You can use the mass spectrometry to detect the elution of this particular protein from the column. And in the analyte, what you will see is you are going to get purification of the ovoalbumine, ovotransferrin, lysozymes, and the ovomucin. Now, whether you are dealing with the plant sources, or whether you are dealing with the bacterial sources, or whether you are dealing with the animal sources, the purification strategies are going to be very fixed. For example, first you are going to take the host cells, depending upon whether it is a plant, bacteria, or animal. You are actually going to first utilize the disruption method. Okay, so first you are going to select the disruption method. You can do the sonications, you can do the thermolysis, you can do the osmotic lysis, or you can do the homogenizations. Then what you're going to do is you are going to get the cell homogenate or cell lysate, right? This cell homogenate, first thing what you can do is you can do ammonium sulfate precipitation. Okay, and that ammonium sulfate precipitation is actually going to give you multiple fractions. It's going to give you like the 20% precipitate. It can actually give you 30% precipitate, give you the 50% precipitate, 65% precipitate, and also a final supernatant. All of these can be analyzed onto the SDS page. So you can actually be able to check the purification of the enzyme on the SDS page, and you can also do the activity of the enzyme. So you can also do the activity of the enzyme, and you can also do the protein concentration. Okay, and that is actually going to be a guiding force whether the purification is going in the right direction or not. Okay, because if you are doing some procedure and if it is giving you no activity, for example, there will be an activity loss, then the purification is not good actually. So then you have to modify the scheme actually. Now, anyway, once you are done with the ammonium sulfate precipitation, what you can do is you can take all these fractions. Suppose you have the protein in this particular fraction. Okay, so if you take this fraction and directly you can take this and perform the HIC. So you can add some more amount of ammonium sulfate, right? Depending on how much ammonium sulfate need you have to add, so that you can actually get the exposure of the hydrophobic patches, and then you can actually be able to perform the HIC. And HIC is also going to give you the different fractions, and all these different fractions can be again analyzed for the SDS page, for the activity, and for the protein concentrations. Now from HIC different fractions, you can just pull the fractions where you have the protein, right, or wherever you have the activity, and then these fractions can be first further loaded onto the ion exchange chromatography, followed by the gel filtration chromatography. And ultimately what you're going to see is you're going to see a purified enzyme if you are going to optimize all these steps in a meticulous way. I don't have examples of discussing all these, but you can actually be able to get an idea, and you can go with this particular scheme. So first you're going to First step, you are going to use you're going to select the disruption method so that the host cell is going to be get disrupted. Second step, you are going to do the amino sulfate precipitation so that you are going to have this suitable fraction where the major amount of activity and the protein concentration is present. And then from this fraction, you can be able to do the hydrophobic interaction chromatography, because the major advantage of the HIC just after the amino sulfate precipitation is that you don't have to waste the time for the dialysis. Because if I want to do the ion exchange chromatography after the amino sulfate precipitation, I have to dialyze and remove the salt, right? So that is not required for HIC. Once you got the different fractions, you can select the fractions, and that selected fraction you can put it for the ion exchange chromatography. And then from the ion exchange chromatography selected fractions, you can take where the activity is very high, you have the minimum number of bands, and your protein concentration is also very high. And then you can put it onto the gel filtration chromatography, and by doing so, all this, you can be able to get the purified protein. If you have the any kind of antibody ready, right, then what you can do is you can take the 65% cut. For example, you have the major activity in this fraction, and you have the antibody which actually can recognize your protein. Then what you can do is at 65% cut, you can actually do the affinity chromatography directly, because most of the time affinity chromatography we should not do from the crude extract, because crude extract is actually going to give you the non-specific binding. So better you do the one round of amino sulfate precipitation so that some of the protein which are not good for the affinity chromatography can be removed. So this is all about the different chromatography technique and the schemes what you can actually be able to follow to purify the enzymes. What we have discussed about the antibody generations in the mice or the rabbits, and then we also discuss about the nickel NTA affinity chromatography also. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss about how you can be able to utilize these enzymes in the different types of applications or operations. Thank you.